FDA approved. Some of them are different phase studies, also for the same for DNA transferring inhibitors. In drug development stages, all these phase studies, initial phases are performed in model systems. Of course, these systems are tested in mice, and, rats, and ferrets, but no new science wants to have more organelle systems and or target cells to be tested in a systematic way, not like whole organism, just a part of organism who has to get these drugs as a target. So in my small testing system, I started these studies in 2015. And of course, I selected best known virus model because such a model study development needs, you have to know the cells very well, the genome, the genomic map, all information possible, and also the virus. And influenza virus is well known, and it is also globally important virus type. It has also 3 different types, and we mostly know the influenza A, and we suffer from it. But these 3 types goes antigenic drift and antigenic shifts, which makes for us a lot of challenge to develop vaccine. Still, there is no effective vaccine for this infection agent. So this is a global problem, and each year seasonally appears both in human and animals. So when you see this pattern of pandemics since 1980s, we have quite a challenge, and they are really smart viruses that they can reassort their genome. What it means, they can quickly shift their genomic types, and our body or a target organ body can recognize, and infection spreads rapidly. But it is for a viral, this is not much complex. There's 11 genes and 11 proteins, and you can manipulate all these proteins by PCR, which is nowadays everywhere available technology, and this can be produced in any in vitro systems, any cell that can grow this virus somewhat. Of course, we are using most productive cell lines to test our systems and doing all sorts of modifications using these genes. Once we introduce a modification, we test in the cell cultures, not in mice or not in rats or human, whatever. But the problem is how we can observe them, because they are tiny organisms, it's not visible. And what the target and why is so important. What we do is in such a systems, here you can see a cellular model. This cell can be long cells, which is targeting bronch cells or pancreatic cells. And we define several mechanistic behaviors, like because virus binds CLS receptor, or virus binds budding, and we use some blockers in cytoplasm, or translation events. Some antiviral molecules target this part, some RNA silencing targets this way, and so on and so forth. However, in host pathogen systems, before designing any disease prediction or prevention studies, you have to understand epigenetic regulations. These days, this is crucial because many of the drug development processes are failing in the latest stage, which causes a lot of enormous economic burden for the pharma companies or governments who tries to do such a R&D studies. But that is also, we suffer from it as a public because we have to use old-fashioned vaccines, which we don't know what is the effect to our body. Actually, the knowledge, this knowledge may contribute to key molecules defining the biomarker and the drug development candidates. So, if we can understand and visualize in a simple systems, for example, cell lines, we can eliminate a lot of failures in our experimental design. So, for example, what type of research methods I tried? After recent years, the Nobel Prize group went to high-resolution microscopy, super-resolution microscopy, and combined with high-resolution immunofluorescence microscopy with my studies. Of course, you can use at the same time drug screening platforms, like if you have a proper cell line, you can infect, and it's successful, you can test different drugs in a tube, in a single tube systems, or multiple tube system with the robotic systems that overnight you can test 100,000 molecules, and you eliminate a lot of false positives. You can screen easily what you want to proceed further. Also, testing protein-protein interaction systems. Testing protein-protein interaction systems is more on the engineering side. You engineer a viral protein, you engineer a host protein, and in the same system, same cellular systems, you transfer them and observe if they get any interaction or not. That's also important because if you target a molecule, for example, here, you have to be able to know that this molecule can interact with this wild negative sense RNA, or here. Any stage, this is important. And further, advanced systems like chipsec, like systems, this provides new alternative, but this is the final stage after developing all the model system, test organisms, or the cells. You can map total chromosome in a nucleotide by nucleotide, so then you can have a more holistic look. So my aim was, if I can observe histone DNA modifications, this is breakthrough research, because there is some histone DNA modification research on cell models, but in a stem cell therapy or other mechanisms, because in infection, this is quite challenging, because once your virus infects cells, it ruptures. The cells die at the end, because your model system doesn't support survival and recovery and all that type of internal responses. But this is what we have to know if we can observe or not, and which changes we can map or not. This is important because then you can go to more organic systems or bigger systems that you know what you look for. The second aim was testing effective epidrugs, because all these epigenetic drugs developed for cancer therapy or immunotherapy is a good target to test if we can see their effect on genome. So this was also an aim. And also I developed in these model systems that I use, I developed protein-protein interaction systems using viral proteins and the host histone proteins, like cellular histone proteins. Are they interacting or not? And later you can map better. Implementing these techniques in infectious disease facilitates better antimicrobial therapies. So I will give you some examples for how we observe histone DNA modifications. Histone looks like this. It packets our DNA. Our DNA is normally cannot fit in a cell, but after histone packaging, they are condensed and they fit in one cell size. So and they have some crucial core histones and some alternative histones. They have different roles in different cellular mesh. For example, here how we do high resolution immunofluorescence microscopy in cell models. This is a bit difficult because histone modification marks, you need some antibodies against these marks. But these antibodies are really low efficient antibodies, and we tested more than 200 antibodies, and we defined 4 acceptable ones that we can detect the signals. These are actually very popular ones. Canine methylation, canine acetylation, like we can show methylation, acetylation, ubiquitination, like very well defined cellular events in the cells using this. So once we studied H1N1 human influenza, very well known type of influenza in laboratory conditions and human lung cells, we follow viral particles in time course, and we observe that after 1 hour, the cells get a virus, it's in the nucleus. So that they do the job in nucleus. So we know that we have to focus on between 3 hours stages. Of course, this experiment went through 24 hours, but later stages are not showing this signaling. So it means to us that we can observe epigenetic marks, but we have to observe if it is happening during this time frame. The first example, same model cells are lung cells, human lung cells, which influenza targets, and we look for repressive mark. Methylation is the repressive mark of the genome. When some part of genome is methylated, that genes doesn't work, it's closed. So these green patterns are the special signals for H2K9 methylation. Means that, of course, this image we quantified using high resolution microscopy, and we observed that on the right down in presentation, you can see 1 hour after infection, we see a quite high methylation. It's very logic because once the virus enters in the cell, it closes cellular control, takes over the control of the cells. It means that if we see some methylation, such as rusting, we have to see acetylation, because if you take a control, you have to open some other parts, which helps you to produce your offspring. Virus needs that, but virus doesn't need the antiviral response of the cells. So this is the acetylation. As explained, acetylation is also being increased. The result of these two black and white picture, what we see is we can really see in the cell model that how virus responds in an early stage, how virus can take over the control of the target cells and start its infection. Also, when you start infection, you need to start specifically. This methylation should not be random, should be targeted. That's why I tried another repressive mark targeting other parts of chromosome. This is H4K23 methylation repressive mark. This didn't show any significant methylation means that, yeah, our methylation is really specific for this virus. It's pattern based, know what it's doing. And also we tested ubiquination, which shows that cellular machinery is still on. Cellular machinery has to be on because the virus needs to produce its genome and offspring. That also very high in the 1 hour after infection. So after all the studies, 2 to 5 years, we showed that we can use cell models, cell lines, bronchoidal or lung cells or any relevant cells for influenza infection to understand how virus targets initially and starts infection. This is very important because whole body in human, you cannot test these differences because in your bronch or lungs, there's multiple factors. There's microbiome. There's also a lot of chemicals. Also, different factors coming from the fitness of the person. So, first we have to define which marks are working in the very sterile condition, very simplistic condition, and these markers can be used to map the genome, and we can know all kinds of tricks of virus, how virus takes control of the human body. Actually, this was histone marks, and then 1 layer down, you have also DNA methylation, because when the histone is open, it's a 1 step, and when histone is open, DNA is exposed. And there should be another layer of control, which is DNA methylation. We also want to test, can we observe DNA methylation as well, the genome or genes target points. So there's 2 types of DNA methylation. DNA methylcytosine, the DNA deoxymethyl. These are a kind of different meaning in de novo demethylation or else related to more functional outcomes for microbiologists. Here, first of all, I tested all cell model, total, not targeting single cell, just total cell lysis. We checked, can we see any changes observable? Yes, we did. It's like similar. Once the cell gets targeted by the virus, direct DNA methylation occurs. That drops 2%. This is very significant because if histone modification occurs and DNA methylation is modulated, means that for us, virus infection can control our chromosomes and the genes to 2 levels. This global transient demethylation is really observed in parallel with the infection start phase of infection.
Otherwise, can you hear me, Dr. Ince? You have only 4 minutes to wrap it up. Could you please finish? Can you hear me?
I couldn't hear the question. Can you write in the chat or say the sound is not really?
He cannot hear me.
Okay.
Dört dakikası olduğunu chatten yazabilir misiniz?
Görüşürüz.
We have what I'm saying. Okay, we could detect actually this type of global events. This is very good news. Also, later stage we tested which type of DNA methylation we have in 2 different models. Because influenza virus is very popular in human, but of course very popular in chicken, in bird flu, and chicken is also one of the global problems. We compare human and the avian influenza. This is human example. In human influenza viruses, we observe bimetal C demethylation, but in not the 5-methoxymetal. In chicken cell lines, chicken model, we try chicken influenza. We observe opposite, like we 5-methoxycytosine is quantification is changed rapidly down, and afterwards even more in hydroxymethyl case. So this is going for technical to basic resulting. All these infections are host specific. Chicken influenza is best in chicken. Human influenza is best in human, but it can change anytime when they expose to each other. For example, if we get the bird flu, we don't get sick in the beginning, but afterwards, after reassortment of virus, we can get a pandemics or else. Like all this information is what I want to give take-home message is influenza virus and basic cell model using lung cells, bronchial cells. We can basically get a lot of information that we don't need using animals. But of course, this needs quite cumbersome R&D stages. But at the end, after showing this 5-year study, we can avoid a lot of mice and rat or ferret use in influenza research, and also such a detailed results are helping drug development stages in a very simple manner. Because here what we show is kind of using cell lines in pharma settings, in industrial pharma settings. They can produce these cell lines and using robotic systems, and overnight they can test 100,000 molecules against this and collect samples and monitor all kinds of changes as we this is possible to imagine genome sequencing or else, and target their studies more precisely. That is all for today. My example. If you have any questions, I can address. Any questions?
Thank you very much, Dr. İnce, for your presentation and contribution to the Congress. Now I would like to introduce our session chair, Serkan Erat, Professor Dr. Serkan Erat. And our session presenters are Arash Ravan Mart, Eda Sezer, and Ruhullah Kianfar. Please welcome them to the stage.
Hocam bu sınıfın içinde online toplantıyı kapatıyorum. Hello, welcome to our today's last session. We have 3 presentations for the final session. Each will be 15 minutes, so we are a little bit ahead of our time. So I'm kindly asking our speakers to adhere the time, not to exceed 15 minutes, but it can be, of course, less than 15 minutes. I think the stage is ready now. Okay, so our first presentation is from the Tebrizina. Araj Javanwart will be present. The level of inbreeding in 4 geographic distinct population of Holstein cattle mirrored by genome-wide DNA profiling. Please go ahead.
Hello everyone, is my voice clear? Okay. Thank you, Mr. Chairman, for chairing this session. Good afternoon, ladies and gentlemen. First of all, before I start my presentation, I think this is the opportune time. I would like to use this opportunity to appreciate from organizer committee to inviting me for this congress. And I want to highlight 2 names. First, Professor Muharram, and second, Dr. Murad. 2 of these people helped me and fasting me. I had a lot of questions and they answered me kindly. And thank you very much for Turkish University for their hospitality and kindness. Okay, let's go to my presentation. The title of my presentation is about the level of inbreeding for geographical different population of Holstein cattle mirrored by genome-wide DNA profiling. For those people, they don't know me, I'm Dr. Arash. My specification is bioinformatics. I spent long time experience travel to different countries, for example, Malaysia, Singapore, and last location I was, Sydney University, bioinformatics in Sydney University. So today I'm gonna to discuss about very sweet story, Holstein cattle and large data analysis. Okay, with this introduction, I'm trying to help organizer and I'm trying to minimize the length of my speech here. So as you know that cattle breeding is very important in our society. We are using different kind of the production from the cattle, for example, milk and meat production. And this picture showing that domestication processes take a long story in human civilization, and also cattle play a fundamental role in our life. I'm trying to specific talking because my professor in University of Sydney told me if you want to speech, you must raise one question and don't go to the theory. You must first try to design one practical question, what you want to do and why you want to do this research. So as you can see here, we have the geographical distribution of the Holstein cattle in different kind of countries. The most of the USA and European country, they have a strategy to increase production of the Holstein cattle. I think in Turkish also we have a dominant population of the Holstein for milk production. And in Iran also in different kind of the province we have Holstein cattle. Most of the farmers, they prefer to breed this breed because this breed significantly carry the long and large scale of the milk production between the other cattle. So as you can see here, Holstein is most important cattle in the life for milk, but we must know both sides of the coin. It means that if we focus to one breed, we have a negative consequences also for this breed. For example, genetic disorder and disease. And because we are using the artificial insemination, it means that we distribute one semen for breeding of the large and 1,000 cows in the different part of the Iranian province, and it caused a lot of problem for us. And if you are not believing my talk, you can see the slides. This slide belongs to the offspring of the most cattle, and you can see that most of the cattle carry high products cattle. They carry the, how to say that, heterozygote genotype for the disease and disorder. And we will try to solve it. So if we use it, if repeatedly we use it, rapidly we use it one bulls for artificial insemination, it make a negative consequences. It means that inbreeding, it may decrease the survivability, less diversity, and blah blah blah. So I'm not going to talk long about because we have a limitation time. I'm not trying to talk more about literature review. Let's to see what I did in my project. So genetic diversity. It is main factor, and immigration, inbreeding, mating system, mutation, genetic drift, and Mendelian sampling, bottleneck, and reproductive biology play the fundamental role to increase genetic diversity between the individuals in one population. So that's why in genetic population we are looking for the DNA level polymorphism between the population and within the individual inside the population. So nowadays, this is a message for those smart students who want to work about genetics and bioinformatics in their thesis or research. Nowadays, most of the European countries, they are using large amount of the, how to say that, DNA information for their modern breeding program. So as our country imposed from sanction from USA, we don't have that technology, but luckily Chinese company and also some of the scientists from USA, they help us for genotyping of our Iranian Holstein population, and I as a bioinformatician, I use that data for analysis. You can see here we have a SNP chip for analysis of the whole genome of the Holstein cattle. You know that we have a 60 pair of chromosomes in the cattle, so 3,000 gene was located inside these 60 chromosomes. And in this project, we genotype all of them for our population to see that what happened inside the genome. So let's go to, I'm trying to escape from this slide because of the limitation of time. Dr. Murad, I'm trying to help you for organization and management because maybe next speaker also want to have enough time. You can see we have a, if we have a SNP chip data, we can go to different kind of analysis. I'm also volunteer. I didn't come to Turkey because of the presentation. Presentation is my second priority. I come to find some professors, some friends to join us, to contribute with us for large amount of the analysis, because neighbor country also play important role for Holstein breeding policy in this country. So if you can see here, most of the scientists in different kind of countries, they are using also same thinking line for genetic analysis in Holstein cattle. So what we should do, know, if we have a large amount of the population and if we have sophisticated tools for analysis of gene, we can analyze them, and based on that we can make a decision what kind of the breeding goal we can use for our breeding program. So let's go to the my goal of analysis. If, as you can see here, I had enough luck to receive a lot of data from different scientists with a different nationality. I have a data. Sorry for interruption. Sorry for interruption. Okay, so different kind of the sample from French, Canada, Poland, and also from Iran. I analyzed them. Why, Professor, I analyzed them? Because I want to know what kind of the company for semen production can help my country efficiently. Because if just you use one country for importation and exportation of the semen, it makes a lot of problem. Because you intentionally, you are going to lose your genetic diversity. That's why that is my goal for designing this project. So, as I said before, I received a lot of data. I sent them for quality control because raw data quality control is very important for us. I analyzed them, and after that, I use a different kind of the bioinformatics tools for analysis of the large data, and now we have a tsunami of the data for genetics. I analyze different kind of the country, Holstein population raw data, and make a conclusion how it looked like. So this is I'm not going to go to further talk because of limitation of time. I just try to say that in population of the genetic, we must produce some population index, and based on that population index, I must go to final conclusion. And you can see here, one of the important parameters is FST. It means that if FST is high, it means that we have inbreeding, and if low, it means that we lost our population inbreeding there. So as you can see here, next generation sequencing data can help us for make a decision globally about our Holstein population. And this is all my result. You can see here different kind of population, Holstein population in different countries. They have a different kind of the advantages. It means that for your own farm, sometimes you must use one company semen, and for second time you must change your company. You must change your policy and use another company. And this is the all analysis data. I don't know if you are familiar or not. This is the GWAS data, genome-wide association data, and it can show that those points exceed from the line. It means that that kind of the gene important for controlling of that investigated trait. And after producing the raw data and raw outputs, you can go to the further analysis, networking system and system biology and also ontology to see what kind of the gene between this significant gene play fundamental role for milk production. And this is some of my outputs. Those circles, they are, how to say that, large circle indicated that that gene is important and you must concentrate or focus in that gene. So blah blah blah. And finally, based on this is the final message of my presentation and last slide. It means that some of the countries, their Holstein, they are has a high similarity between the Iranian Holstein and that countries Holstein. What does it mean? It means that you must avoid importation of the semen from that country, and you must put for those companies their Holstein genomes far from our genomes, because if you are using that kind of the cattle, genetic diversity of your own Holstein cattle will be increased. And that's all. Thank you very much for your patience and attention to my presentation. Again, if you have any question, please welcome. I'm ready to answer your question. Appreciated, Chairman, from your patience and not stopping me for my presentation.
Thank you, Javan. Do you have any question for Javan? Thank you. I want to also thank you special for you. To say good words about our university and me. So thank you, Javan. And I would like to invite Eda Sezer for second presentation. Okay. Second presentation.
Hello everyone, can you hear me clearly? So, my name is Eda Sezer. Today I'm going to talk about my PhD project, which I did at University of Southampton in England, UK. So, in my PhD, we were trying to look for the impact of maternal high-fat diet consumption during two specific periods, pre-implantation and whole pregnancy lactation periods on offspring behavior. So here is my outline. First, I'll give you some background information. I'm going to talk about Barker hypothesis and developmental origin of health and disease concept that we used in our project. Then I'll try to explain the importance of these two periods. And then I'll tell you the importance of high-fat diet and its relation with obesity. Then I'll explain in the literature information how the maternal high-fat diet consumption affects the offspring brain. Hopefully, I'll explain our mice model and give you some behavioral data. First, I want to mention David Barker. He was actually one of the reasons that I chose Southampton when I was looking for my PhD. He actually one of the most influential epidemiologists for our times, and he was working the data in the poorest region of England. And he found a correlation between prenatal nutrition and later on dying from cardiovascular disorder. And he suggested that fetal undernutrition, especially in middle to late gestation, resulting in disproportional fetal growth and characteristics such as small size at birth. And this later programmed the cardiovascular disorder in their future life. And later on, Barker hypothesis named differently the fetal origin of adult disease, and then later on they named this theory developmental origin of health and disease concept. So basically, this concept addressing how nutrition during critical window of development affecting offspring future health. So we choose two periods. The first one is pre-implantation period, which is the first 3.5 days of the mouse pregnancy. It starts with the zygote formation to the blastocyst formation. And it's really important because self-weight decisions are actually happening during that stage. And we know there is no direct link between moms and offspring, but somehow embryo can sense the altered environment and react to do that. Our second period that we choose in our project, whole pregnancy and lactation period. And we know moms provide the first environment for the offspring, and this is there is biocommunication between mom and offspring via placenta and milk. Maternal substance such as peptides, hormones can pass through the moms via placenta and milk. And if there is altered environment during those periods, and somehow embryo can adopt this, adjust themselves this altered environment, and to be able to survive in a less optimal environment. But you know, David Barker and his colleague was working with the data coming from a family study after the post-era. But if you look at now, it has less relevance in our world because we are suffering from obesity. And if you look at the data, there are 2.5 billion people are suffering either being overweight or obese. And this is not just for adults, also children are suffering from that. And for our project, we are looking for the mom side of the story. And for example, if you look at the data, obesity is actually the 25% of the pregnant ladies in the UK. So, and what is obesity? It's actually excessive fat accumulation in the body, and consumption of the high-fat diet is one of the main reasons. And diet containing the more than 30% of total energy is the reason for development of obesity in human. But it's so difficult to work with the human data. That's why we are using the marine models. And if you look at the literature, animal studies, if you want to work on obesity, it's changing 30 and 78% of total energy intake in terms of fat. And we know from the literature, and there are a lot of research saying obese mom children and high-fat diet induced obesity studies are showing that offspring coming from those moms have increased risk for depression, attention deficit and hyperactivity disorder, anxiety, even schizophrenia. And this disorder is not only affecting the people, it's also affecting the society. So considering all those factors, we wanted to identify the possible effects of maternal high-fat diet consumption during those periods, and we are not making an obesity model. Remember the David Barker, because they were working only the confined period, and we just want to analyze how these going to impact on offspring learning, memory, and social behavior. And we wanted to characterize the sex differences in those measured parameters. First, I'd like to explain our mice model. We are using MF1 outbred species, and we have three different groups. The first one is the control group. We are feeding the moms, pregnant moms, 12 standard diets, which is the 7% of kilocalorie of fat. Our second group is the embryonic high-fat diet group. We are just feeding the mom with the high-fat diet during only pre-implantation period, and then we switch to their diet to the control diet. And our third group is the high-fat diet groups. We fed the moms with the high-fat diet during whole pregnancy and lactation periods. Then moms give birth, and you know it's so difficult to arrange the offspring number. So that's why we normalize the offspring number to the 6 to adjust their lactation demands. We left 3 males and 3 females per group. Then during the lactation period, which is the 3 weeks in the mice, then we wean the offspring from the moms, and we started feeding them with the 12 standard diet. Then they have 15 weeks of life periods, and we weight them in a weekly basis. Before I explain my result, it's a continuous study, and my colleague Diego, he was working with the same model, and he actually finds so many data, but the most important thing that we want to focus on the behavior part, he found there's altered neurogenesis in the offspring coming from, especially male offspring, coming from the high-fat diet months. So we decided to testing the offspring starting with week 4 until the week 15. So my colleague, Irene, we were working on the same animal model, and we started testing the offspring. I don't know, can you see that the blue lines, I highlighted them, made by me, the rest, she was responsible. So I'll explain each test individually, and I'll give you the results later on. So we started testing them with the open field test, novel object recognition test, elevated plasmase, and their adolescent period, and then we repeat those tests when they reach adulthood. And also we did the teammates and social interaction just once. And at the end of the 15 weeks, we collected all the tissues. So here's a result. You can see the males and females offspring body weight changes throughout their lives. If you look at it week 2, 3, and 4, you can see offspring coming from high-fat diet groups, they were actually heavier than the control. And the rest, especially males offspring from embryonic high-fat diet group, they didn't show anything differently. But the females offspring coming from embryonic high-fat diet group, they were actually thinner than the control in their adulthood periods, such as in week 9, 12, 13, and 14. So we kept testing them in open field tests. What is this test? We are using to evaluate their locomotor, explorative, and anxiety-like behavior. So we did this test in week 4 and 10. We just tested the offspring for 10 minutes, and we are using a software to track the animal. So software gives us the whole distance traveled, ambulatory count, the speed, resting time. So basically, we are just putting the animal inside of this base and then letting them to explore and doing those behavior. And then we analyze the result as first and second 5 minutes. And we use a hierarchical linear models for the comparisons. And if you look at the data results, you can see that males offspring coming from embryonic high-fat diet group, they're actually traveling less, resting more, and moving less compared to control, and they're jumping less. Their both locomotor and explorative behavior changed. And also, if you look at the female side on the right side, left side to me, to the table, you can see vertical count, which is a behavior the animal just standing up position in their hind legs. And females offspring coming from high-fat diet months, they're actually doing this behavior more than control. Something is changing. And then when we repeat this test in week 10, we didn't see any significant differences in those parameters. Then, if you are working with animals, one test is not enough. You have to prove your results with another test. And then we did elevated plasmase. Basically, we have 2 closed arms and 2 open arms. Usually, animals, mice specifically, they wanted to. There is a behavior called hematoxis. They want to touch the wall. They feel more safe in those places. And if they are spending more time on the closed arm, that means they are showing anxiety-like behavior. And at the end, we calculated the arm entry and time spent in those arms. The only the statistically significant differences we found, high-fat diet, females offspring, which is showing the, again, right? They are actually doing, entering more in the closed arm. But when we repeat those tests in week 12, we again didn't see any significant differences. Then we kept testing offspring to measure their hippocampal memory, which we did this test in week 4 and 10. Basically, we are using the same platform, using 2 similar objects in the familiarization phase, testing them for just they are getting used to the platform and this object, and then taking the animal, cleaning those places, then putting a novel object on the platform. What we expect, they should spend more time with the novel object, they should remember. And then we are, again, assessing their short-term hippocampal memory. And then we are calculating the investigation time and discrimination index based on this phase and familiarization phase. So if those index are higher, they are showing better memory. But we didn't see any significant differences, those measured parameters, neither in week 4 nor in week 10. Then we did a T-maze, which is another test to evaluate their short-term spectral memory. Basically, we are putting the animal in the base site, letting them choose the one arm, restricting them in those arms, then picking up animal gently, starting again, and then they should go to the unvisited arm, which is a way they should remember they go to the previous arm, and then based on that, we are calculating an alternation score. At the end, we did in this test in week 8, but we didn't see any significant differences in their alternation scores. Finally, is our test we also measured their social behavior, which is a test we did in week 12, which is their early adulthood period in mice. So basically, we are using the same platform that we use in open field tests, and then we are using completely novel mice, and then we are evaluating their behavior. Our tested mice, how it's going to behave is this novel mice. We are categorizing their behavior in 4, and for example, if because mice are very social animals and they want to know who are encountering. For example, they are sniffing time, body sniff, nose to nose sniff. These are evaluate under the social investigative behavior. And for example, affiliative behavior, we use the social grooming as a sign. But instead of exploring the novel mice, if they spend more time rearing on the wall or just doing upright position in the platform, which is a sign for the non-social behavior, or sometimes they can claw or pinning these novel mice. So all this, it was really tricky to analyze the social interaction test, but we might be able to do it. And then if you look at the results, we didn't see any significant differences, all those parameters, neither in their sniffing times or their affiliative behavior, or in their non-social behavior. Offspring coming from this diet treatment group, they were different than the control or each other. As a summary, so in the current study, administration of the high-fat diet to the pregnant mice during either only pre-implantation or whole pregnancy and lactation period affected the offspring growth. And you might remember the juvenile offspring coming from high-fat diet moms, males and females offspring, they were heavier than the control. And then, if you remember the anxiety-like test results, especially males offspring coming from the embryonic high-fat diet groups, they actually showed some anxiety-like behavior. And then, high-fat diet, the females offspring coming from high-fat diet moms, they were also showed some anxiety-like behavior. But memory functions and social behaviors were not affected by maternal high-fat diet consumption. So what is actually happening in the offspring? So one way to explain this, those periods we choose, they're actually really important for creating the genome, and the epigenetic alteration happened during these times. And that might explain those results. And you might remember from the schematic representation, we started feeding the offspring with the trough diet. Yes, we started testing them in week 4, and during the whole lifetime period, we started feeding them with the trough diet. Maybe this diet, consumption of the trough diet, maybe lifted those previously seen results. So we can say that offspring locomotor and exploitative behavior, they actually time and sex-dependent manner we show the results. And exposure to normal diet can rescue the phenotype in adulthood. So I'd like to thank, finishing my presentation, first the biomedical research faculty at University of Southampton. We did this project during the lockdown, and we spent 1.5 years, and they were really helpful for us. And I'd like to thank my wonderful supervisor and my colleagues, and also my sponsor, Turkish Ministry of National Education, and thanks for listening. And if you have any question, I'm happy to answer them.
Thank you, Suzanne. Any question for me?
Yok question. Actually, thank you very much for your outstanding speech and your beautiful product of this research. And I just have one statistical advice, because as I enjoy from your outputs, it seems that most of your result is non-significant, and non-significant result is not related to your nature of the study. Maybe, perhaps, I'm not sure about that. It is related to some of your raw data inside the treatments. I mean that variation inside your treatments is carried. Some of them is outlier. How to say that? Maybe if you go to normality test,
yeah,
because from the bar chart, it makes sense for me. Most of the mouse, because they are different sex,
yes,
male and female, they didn't respond. They didn't show similar response to one treatment, isn't it?
So,
am I right?
Yeah, well, I'm getting that comments in most of my presentation.
My summary of question is standard error is too large.
Yeah,
that's why it is non-significant.
Yeah, the thing is, we are using the hierarchical model, including the liter size and body weights into the analysis. So to be able to analyze the data in that method, we are using the Z conversion. So that's why we don't need to normalize the data. So that's fine. But when I'm, yeah, you're welcome. Yeah.
Thank you, Jawan and Cesar. Thank you.
You're welcome. Thank you for listening.
I would like to invite Kian for his presentation, last presentation.
Hello, everybody. I am Kian from University of Tabriz. Thank you for your hosting and organizing committee for this congress. My object is effect of probiotic and propolis supplementation with different xylanase enzyme on equality in layer and fed basal diet, wet-based basal diet. Wheat is one of common cereal in poultry and soy diet in worldwide. However, wheat contain 5 to 8% of arabinoxylan, which compound of mono saccharide, arabinose and xylose. Arabinoxylan as a main compound of the wheat cell wall exhibit anti-nutritional factor, anti-nutritional effect, similar to beta-glucan in barley. Non-starch polysaccharide, such as arabinose, is a large molecule that absorb or trap water and increase the viscosity of intestinal digester. This evaluated viscosity reduces the diffusion of digestive enzyme and nutrient, and finally reduce absorption and digestion of nutrient. Soluble NSP encapsule nutrient and disrupt gut physiology, leading to reduced nutritional digestibility and lower metabolizable energy and increased feed conversion ratio. Propolis as a potential solution for anti-nutritional factor of NSP. Propolis are now also known as a B group, is a resinous component collected by honeybee from plant buds and leaves.
The composition of propolis is very different based on region and honeybee race, but it's contain resin, wax, fatty acid, oleic acid, pollen, trace mineral and organic component, vitamin, and some of mineral. Resin contribute flavonoid and phenolic acid with unknown antimicrobial and antifungal property. Propolis potential to mitigate the negative effect of NSP based diet due to its bioactive component, which may enhance gut health, nutritional digestibility, and overall bird performance. To this aim, we evaluate the synergy effect of propolis and probiotic supplementation with different level of xylanase enzyme on equality of laying hen. For this purpose, we use the experimental design three multiple tree factorial arrangement with 9 treatment diet. This experiment was conducted in the research farm of Lake Hens in the University of Paris. You can see a picture of this farm. Treatment include 9 treatment. We have for investigate the effect of propolis and probiotic on NSP effect in lake. Fit ingredient and composition of the control diet that is added enzyme and propolis and probiotic to this basal diet for lake hen. Traits measured during this experiment include shell quality, internal quality of egg, and production performance such as feed intake, egg production, FCR, egg base, and other things. Tools and methods for measuring egg quality shown in this slide as a picture. You can see this slide. We use cryogenic milling for converting resinous propolis to powder. In previous session, Dr. Besharati said about propolis, and he used butter extract propolis. But we use propolis, complex propolis as a food. For this reason, we freeze propolis in nitrogen, liquid nitrogen, and then milling to converting resinous propolis to powder. Result and discussion was additive propolis and probiotic significantly increased production percentage, show this slide significantly, and enzyme had a significant effect on production performance, production of egg, but have a difference between level of enzyme and 500 ppm enzyme have a better function in this trait. The interaction between enzyme and additive was significant, and best treatment in this session is 5% enzyme plus propolis that have a more product compared with control group. Propolis significantly increased weight of egg compared to the control group as shown in this slide. The enzyme level also had a significant effect on egg weight, but egg weight and increased egg weight in 500 ppm enzyme and propolis. The interaction effect of propolis and interaction effect of additive and enzyme was significant on egg weight and same as egg production. Treatment contained 500 enzyme and propolis had a better effect. We mentioned half unit. Half unit is a system for analyzing internal quality with using equation half units. We can measure it to evaluate internal quality. As shown in this slide, the probiotics significantly reduced half unit compared to control group, but propolis increased half unit significantly compared with probiotic diets. Enzyme had a significant effect on half unit and increased half unit by using 500 ppm enzyme in diet. The interaction between enzyme level and additive significantly affected on half unit and same as previous steroid. Better treatment was 500 enzyme and propolis together. The yolk color index measured with fang color rush index. Propolis significantly increased yolk color compared to the control group, as shown in this slide. This effect due to pollen in propolis. Propolis contain some pollen and pollen contain carotenoid. Carotenoid can increase egg yolk color. Enzyme have a significant effect in this trait and significantly increase egg yolk. The interaction between enzyme and additive was significant, and best treatment in this trait, same as other trait, was 500 ppm enzyme and propolis together. The effect of adding propolis and probiotic on eggshell quality or egg thickness was not significant, and all of treatment have the same thickness of eggshell. We measure a specific gravity for analyzing egg quality and some of the internal quality with salt solution. The type of addition and additive and had no significant effect on a specific gravity, but enzyme have a significant effect on a specific gravity, and enzyme significantly increased egg specific gravity due to increasing the digestibility of nutrients and absorption of nutrients such as calcium, phosphorus, magnesium, and other minerals. Conclusion: this study demonstrated the potential benefit of propolis and probiotic as a dietary supplement for laying hen vet-based diet. Once additives significantly enhance egg production percentage compared to the control group, with propolis showing a more pronounced effect on increasing egg weight and yolk color. Enzyme supplementation, particularly at 500 ppm, consistently improved egg production, egg weight, and yolk color, while also enhanced specific gravity, indicating better egg quality. The interaction between enzyme level and propolis further highlighted the synergic potential of this treatment in optimizing yolk color and egg production. Overall combination of propolis and probiotic with xylanase enzyme can be effective strategy to mitigate the anti-nutritional effect of waste diet, improving bus production performance and certain aspects of egg quality in laying hen. Further research is recommended to explore the long-term effect and underlying mechanism of this interaction. Thank you for your time and attention.
Thank you for your presentation. Any question for?
Hi, thank you for your presentation. My question is, how feasible to be able to use this technology? Like, if you consider the cost and everything, you know, because obtaining the propolis and adding them the enzyme or supplements into the increasing the quality, how feasible? Yeah.
My result is applicable for farmer, and farmer can use my advice to increase the quality. Cost, commercially propolis same price with enzyme and not very expensive. But when we do this experiment, propolis very low cost, but now propolis increased in price.
Thank you.
Thanks. Thank you, Cameron.
I would like to thank all our participants for their presentations. Now, our session chair, Murat Er, will share their certificates.
Orada değil.
Masa bitmiyor başkanım.